Hello students. Today we're going to discuss about Unit 12B, infectious parasitic diseases, protozoa and metazoan parasites. This is from MSc, second year. So in this chapter, we're going to discuss about the protozoan diseases like coccidiosis, enterococcidiosis, whirling disease, ichthyothireus, metazoan parasitic diseases like dactylogyrosis, gyrodactylosis, diplozoan disease, and sanguinocoliasis, diplostomiasis. Now, the first disease that we are going to discuss is ichthyobodosis. Now, we are going to see what are the causes, symptoms, and the type of treatment that we give for this. Now, ichthyobodosis, it is a fish disease caused by Ichthyopodonecator. Now, this is a parasite responsible for this disease and which is present in freshwater fishes. The early name of this disease, we also call it as Costionecatrix. Now, this is regarded as primarily as a freshwater fish ectoparasite with a broad host range, and it is a protozoan parasite. This was first described by L.F. Hanigay in 1883 and more fully in 1884, and he named it as Bodonecator. Now, let us see the characteristic features of this Ichthyobodonecator. These parasites are microscopic sizes, vary from 5 to 6 micrometers. Ichthyobodonecator, it often found in infesting fish's skin and gills, including salmonids. These parasites can reduce the ability of young salmon to adapt to seawater if it invests in the gill, and then free swimming form is oval to spherical, and it measures from 5 to 8 micrometers. Its pre-division stage has 3 and 4 flagella, and larger stages have 2 flagella. These flagella, they get attached with the host by its pointed anterior end. Now, in adverse environmental conditions, they form a cyst, which becomes an additional infection source. They process a ventral flat disc. Now, we'll see this fish species affected by Ichthyobodonecator. They have recorded more than 60 different host species in both freshwater and marine water. Few examples like Atlantic cod, salmon, trout, Cyprinus carpio, haddock, tarbot, halibut, spotted wolfish, Clarius garipennis, flounder, mullet, and tilapia. Mostly, we find them in juvenile fish, that is fry and fingerlings, and adults are believed to be particularly susceptible to the attack. Now, how their life cycle is going to be. So, the parasites that attach to the body by attachment organ, and the body is pyriform. Its free non-feeding form is oval shaped, which has 2 or rarely 4 flagella. The parasitic stage found in fish bodies is known as trophont. Now, this parasitic trophon develops a suitable size, drops off from the host body, becomes a cyst, and turns into a tomont stage. Within the cyst, tomont divides into 10 to 11 divisions to produce small tomites. Tomites break the cyst wall to become theronts. These theronts are the infective stages that affect the host. Now, the symptoms are going to be like this. These are frequently languid and listless and typically exhibit flashing behavior. In more advanced cases, they are characterized by a blue-gray film on the surface of fish owing to elevated mucus generation and hyperplasia of the epidermal epithelium. Gill hyperplasia and lamellar fusion are signs of ichthyobodosis, a disease caused by infestation of the gills by parasites. Secondary bacterial and fungal infections are common in fish with ichthyobodosis. This is how the infected fish look like. Now, how do we diagnose this? The diagnosis is done by, we can see a parasite protozoan that infects fish. It can be definitively made through various methods. So diagnosis is done by various methods. One common method is through wet mount skin and gill preparations, where the parasite can be observed under a microscope. This typically exhibits an asymmetrical oval body in its attached form, with fewer free swimming forms that are more ellipsoidal in shape. Sometimes two unequal flagella can be seen from the anterior end, which lie along a funnel-shaped groove on the ventral side of the parasite. In stained histological sections, the attached forms of the parasite can also be detected. There are certain characteristics that can aid in the diagnosis of ichthyobodonecator. For instance, when the parasite turns its crescent-shaped body, the free form of the parasite may exhibit flickering motion. This is the main symptom. Now, how to prevent this? First and foremost, providing a stress-free environment for the fish is crucial in preventing ichthyobodosis. This can be achieved by minimizing environmental stressors such as sudden changes in water temperature, poor water quality, overcrowding, and inadequate nutrition. Maintaining stable water conditions and providing appropriate tank or pond size for the fish species are essential to keep the fish healthy and less susceptible to diseases. Regular cleaning of the aquarium, pond, or tank is also important in preventing the buildup of organic debris that can serve as a breeding ground for parasites. Apart from this, excess food, uneaten food, and fish waste should be removed promptly to prevent water contamination and the spread of parasites. Now, what are the proper feeding practices that can maintain in preventing the disease? Feeding the fish the correct amount of food and avoiding overfeeding can prevent excess organic debris in the water, reducing the chances of parasite infestation. Avoiding overstocking of fish in a tank or pond is another preventive measure. Overcrowding can lead to increased stress, poor water quality, and higher chances of disease outbreaks. Ensuring that the fish population does not exceed the capacity of the holding tank or pond can help prevent the spread of diseases like ichthyobodosis. Now, one more method is quarantining. Quarantining new fish before introducing them to an established tank or pond is also recommended. Now, quarantine periods allow for observation and potential treatment of any potential diseases or parasites that may have been introduced with new fish, preventing the spread of diseases to the existing fish population. Now, how to treat this infected fish? Formalin 166 ppm for 1 hour bath, that is 1:4,000 for 15 to 20 minutes is well tolerated. Treatment for 1 hour with formalin 1 is to 6,000 is also very effective. Ponds with infected fish are drained and quicklime, chloride, or limes are added before restocking. Exposing fingerlings to a weak solution of malachite green, that is 1 is to 4 lakhs for 40 to 60 minutes, is also advisable. Cyprinids lose their infection when dipped daily in copper sulfate, that is 500 ppm, for 1 to 2 minutes, or salt solution, that is 5%, are recommended for larger fish. Other chemicals like methyl blue, potassium permanganate, Lysol, etc. can be used. Now, let us discuss about the other disease called as whirling disease. So, what is this whirling disease in fish? It is caused by a microscopic parasite called Myxobolus cerebralis. This parasite primarily affects a salmonid fish such as trout and salmon. The disease is primarily transmitted through direct contact between infected and susceptible fish or through contaminated water sources. Myxosporians are a group of parasitic protozoans that belong to the phylum Myxozoa. Myxozoa is a species group of endoparasitic cnidarians that can cause severe ecological and economic effects. They are commonly found in aquatic environments, especially in the marine ecosystem. They are mostly pathogenic to both wild and farmed fish. Now, this is the life cycle of this whirling disease. Now, the life cycle of the parasite involves two hosts, a tubificid form, worm, that is a worm, and a salmonid fish. The parasite begins its life cycle as spores released by infected fish. The life cycle of whirling disease begins with microscopic spores present on the river bottom. These spores are consumed by bottom-dwelling tubifex worms. Within the tubifex worms, the spores undergo a transformation and become triactinomyxons. This stage is capable of infecting fish. When the infected tubificid worm is consumed by a susceptible salmonid fish, the triactinomyxon is released and enters the fish's body. The parasite then invades the fish's cartilage, particularly affecting the cranial and spinal cartilage. When the infected fish dies and decomposes or is consumed by another predator, the spores within its body are released into the water, initiating the cycle, a new cycle. The life cycle of Myxobolus cerebralis, a parasitic organism, it involves several stages. Now, if you see the stages, stage 1 it is ingested by tubifex, and this myxospore develops within the oligochaetae post tubifex. Stage 2 has the popular filaments are extruded, and this cerebralis spore anchors itself into the gut epithelium. The shell walls open, allowing the binucleate sporoplasm to escape and penetrate between epithelial cells. Stage 3 involves The binucleate sporoplasm undergoes interepithelial schizogonic multiplication. Stage 4 involves the sporoplasm transforms into uninucleate one-cell stage. Then stage 5 involves two uninucleate cells fuse through plasmogamy, resulting in the formation of a binucleate cell stage. Then stage 6, these both nuclei within the binucleate cell undergo mitotic division, giving rise to a 4 nuclei stage. Then plasmatomy occurs, forming a four cell stage where two cells begin to develop the other two cells. Through three mitotic divisions of both generative cells and two mitotic divisions of the somatic cells, six gametocytes enveloped by 8 somatic cells are produced. Then, meiotic division of the 6 diploid gametocytes leads to the formation of 16 haploid gametocytes and 16 polar bodies. Then, copulation of each pair of alpha and beta gametes results in the reproduction of 8 zygotes. After two mitotic divisions of the zygote, three pyramidically arranged cells and one inner cell form, leading to sporoblast formation. Then stage 13 includes the three peripheral cells undergo mitotic division, producing three capsulogenic and three valvogenic cells. Then next stage is the valvogenic cells extend around the capsulogenic cells, while internal cleavage of the developing sporoplasm cell produces one generative cell enveloped by one somatic cell. The sporoplasm remains naked in the pansosporocyst until reaching the final number of germs through repeating mitotic divisions. Then the mature triactinomyxon spore inflates, completing the life cycle of Myxobolus cerebralis. Now, this is how the life cycle looks. These are the stages that we see. See how the parasite invests, the invasion, that is the how it gets attached to the fish. Then schizogony, gametogony, and the sporogony, how the spores are released, and again how the new life cycle starts. Now, how to prevent this whirling disease? To prevent the spread of whirling disease, it is essential to clean clean waders, boats, paddles, and any equipment that comes into contact with water, ensuring they are free of mud or plant matter. Now, how to treat this disease? Naladixic acid can be an excellent product for addressing symptoms that mimic whirling disease in aquariums, particularly in cases where true whirling disease is not present. Combining naladixic acid with products like metronidazole or medicated wonder shells, especially for discus and other cichlids has proven to be effective. Now we are going to discuss about another disease, ichthyotherius. What exactly is this? This is a parasite protozoa that causes ichthyotheriasis. This is more commonly known as white spot disease or simply Ike. So this is an obligate parasite, meaning it must have a fish host to survive. Although parts of its life cycle are spent in the open water, this protozoan has multiple life cycles. The most notable being the phase where it causes visible white cysts. Ike spreads quickly and, if left untreated, will lead to 100% mortality in fish. Now, the life cycle involves, in order to prevent and treat Ike, we need to first understand its life cycle. The three distinct life stages of Ike are the tomont, theront, and profont. So, in this, again, there are different stages. Exciting tomont, tomont cyst, dividing tomonts within cyst, infective theronts leaving cyst, dividing tomites within the cyst on fish. Now, this is the life cycle, how it looks. Now, the symptoms include fins clamped next body and then covering at top or bottom of tank, congregating around oxygen sources, rubbing or scratching against surfaces, flashing, reduced appetite, short choppy gill movements, white spot cysts. These are the symptoms we can see in this. Now, how to prevent this? It goes without saying that Ike is some pretty nasty stuff. The best way to deal with it is to never get it in the first place. So prevention is the key for this disease. The single best way to prevent Ike in our aquarium is to quarantine all new fish or live plants. This will require an extra aquarium, but it need not be large or extravagant. There are a wide variety of diseases that can hitch a ride on your new fish or plants. For this reason, it is best to quarantine them for at least 4 weeks, but longer is better. During this time, most diseases should reveal themselves. With live plants, it's best to buy them from tanks without fish. If they have never been exposed to fish bearing water, Ike is much less of a concern. The most common medications are malachite green with hyaline blue, formalin, and copper-based medicines. Popular products will often mix these primary ingredients with others in proprietary blends that will solve the problem. Next, we are going to discuss about dactylogyrus. It's also called as gill fluke disease. Now, what are the causes, symptoms, and type of treatment? We'll discuss now. As an avid fish enthusiast, it's important to be aware of the common parasitic infestation called gill flukes caused by dactylogyrus that can affect fish health and well-being. Now, what are the causes? As it is a monogeneous parasite, commonly infects the gills of marine and freshwater fish. Dactylogyrus species is a group of monogenean gill parasites that are highly specific to freshwater fish of the family Cyprinidae. It's a highly diverse genus with more than 900 described species that primarily parasitize the gills of Cyprinidae. They are ectoparasites with a direct life cycle. Now, these flukes have distinct characteristics that include a dorso-ventrally flattened body with bilateral symmetry resembling a leaf shape and measuring around 7 to 8 mm in length. They have 2 pairs of anchors, 4 eye spots, and 7 pairs of marginal hooks. The structure of gill flukes also includes 1 or 2 connective bars and 2 spindle-shaped needles in their seminal vesicle. They possess an oral sucker around their mouth and a ventral sucker for attachment, known as a hapter. Gill flukes are hermaphrodites, meaning they have both male and female reproductive organs, and they are oviparous, laying eggs for reproduction. The alimentary canal of gill flukes is well developed with a muscular pharynx and esophagus for feeding. They also have flame cells, which serve as excretory organs. These characteristics make gill flukes a complex and specialized parasite that can cause harm to the gills of fish, leading to various health issues. Understanding the anatomy and biology of gill flukes is crucial in identifying and treating the infestations in fish populations. Now, let us look at the life cycle of this Dictylogyrus species. It involves one host, the fish, and is relatively simple compared to other parasitic infections. Here is a more detailed explanation of the life cycle. Now, the first stage is egg stage. Adult Dactylogyrus parasites attach themselves to the gills of a freshwater fish and lay eggs. These eggs are attached to the gill filaments or the surrounding tissue. Then the next is Onchomyracidium stage. The eggs hatch into free-swimming larvae called Onchomyracidia. This Onchomyracidia has a ciliated epidermis and is equipped with hooks and suckers that allow them to attach the fish host's gills. Next is adulthood stage. This Onchomyracidia attaches themselves to the host fish's gills and develops into adult parasites. The adult Dactylogyrus parasites feed on the host's blood and tissues using their hooks to anchor themselves to the gill filaments. Next comes to reproduction stage. The adult parasites continue to lay eggs, which either remain attached to the gill filaments or are released into the water. Next is the completion stage. The life cycle of Dactylogyrus species is completed when the eggs hatch into Onchomyracidia, which then attach to new hosts and begin the cycle again. So overall, the life cycle of Dactylogyrus species involves only one host, the freshwater fish, and can be completed relatively quickly, leading to heavy infestations in fish populations if not controlled. Next, let us see the symptoms. The fish may become restless and swim towards the water's surface in an attempt to obtain oxygen, as they may struggle to respire properly due to gill damage. The gills themselves may become swollen and pale in color, and there may be an increase in mucus secretion, leading to a high mucus coating on the gills. The fish may also lose weight and appear thin, and the edges of the gills may take on a grayish hue. In severe cases, the fish may exhibit open operculum as they struggle to breathe. Dark coloration, often appearing as patches or spots, may also be noticeable among the diseased fish. Affected fish may lose their appetite and cease to feed, and may exhibit abnormal behavior such as swimming near the water surface and jumping out of the water in an attempt to obtain more oxygen. Now, how to diagnose this disease? Gill flukes attaches to the host gill epithelium with hooks on their posterior where they feed on mucus and blood. Infestations may lead to secondary infections with other pathogens. The diagnosis of this Dactylogyrus species infestations in fish can be done through different methods, including physical observation. The first one is physical observation. Physical observation is needed to observe any disease. Gill smear. The first method is gill smear. So in this, remove part of a gill arc with scissors and holding the tissue in forceps, smear the lamella on a microscope slide. The material adhering to the slide may contain mucus, epithelial cells, and blood cells of the gills. Allow the smear to air dry and pass the slide through an open flame 2 to 3 times, fixing the smear on the slide. If parasites are found, it will compare with the characteristics of Dactylogyrus species. Then it will be sure what sort of parasite caused the disease. Then the macroscopic examination. The presence of Dactylogyrus species can be seen through the naked eye. The adult parasites appear as tiny white or yellowish specks, often clinging to the gills of the fish. The presence of brownish black spots on the gills or bleeding and reddening on the gills and body of the fish also indicates an infestation. Now, how to prevent this gill fluke? To prevent gill fluke infestations in fish, several measures can be taken. Maintaining good water quality and pH. It involves regular monitoring and maintenance of water quality parameters such as temperature, pH, ammonia, nitrite, and nitrate levels can help create an environment that is less conducive to the proliferation of fish parasites, including gill flukes. Increasing water exchange. Frequent water exchanges, either through partial water changes or continuous water flow, can help dilute and remove potential sources of gill fluke infestation, reducing the risk of infection. Then reducing organic load. Excess organic matter in the water, such as uneaten food, decaying plants, and fish waste can contribute to poor water quality and provide a breeding ground for parasites. Regular removal of debris, proper waste management, and avoiding overfeeding can help reduce the organic load in the water and minimize the risk of gill fluke infestations. Then, liming. The application of lime, that is calcium carbonate, to the water can help raise the pH and create a less favorable environment for gill flukes to thrive. However, the appropriate dosage and application method should be carefully followed to avoid any adverse effects on fish or other aquatic organisms. Now, how to treat these gill flukes? It's a type of parasitic flatworm that commonly affects fish in aquariums and ponds, and it can cause several health problems, including damage to the gills, loss of appetite, and stunted growth. So, so these are the type of treatments that we can give. First one is salt bath. The one effective treatment for dactylogyrus is a salt bath. Dissolve aquarium or pond salt in water and place your fish in the solution for several minutes. The concentration of salt should be based on the type of fish we have. So it's best to consult with a veterinarian or experienced fish keeper for the appropriate doses. Next one is formalin treatment. Formalin is a remedy in many cases used to treat parasitic infections in fish. Now follow the dosage guidelines carefully and examine the fish intently for any damaging reactions. Then next is improve water quality. Poor water can make contributions to the improvement and unfolding of dactylogyrus infections. Maintain correct water. First rate 9 with the useful resource of performing ordinary water changes. Holding the water pH within the amazing range and making certain enough filtration methods. Next, we're going to discuss about another disease, Gyrodactylus. This is a type of parasitic flatworm that infects fish. This parasite is microscopic and can only be seen under a microscope. It is an ectoparasite, which means it lives on the skin, fins, and gills of the fish. Gyrodactylus is highly contagious and can spread rapidly in a fish population. Gyrodactylus infections can occur due to a variety of factors. Fish that are stressed, injured, or have poor water quality are more susceptible to infections. The most common way that Gyrodactylus is spread is through direct contact between infected and non-infected fish. The parasite can also be introduced to a tank through contaminated water, plants, or equipment. Let us see the symptoms of this infection. The symptoms can vary depending on the severity of the infection. In mild cases, fish may display no symptoms at all. However, in more severe cases, fish may show signs of stress such as excessive mucus production, rapid breathing, flashing, flicking, rubbing against objects, red inflamed gills, damaged fins, and discoloration of the skin. Now, how to identify these infections? These infections can be identified by examining the skin, fins, and gills of the fish under a microscope. If you're not experienced in identifying parasites, it's best to consult with a veterinarian or an experienced fish keeper. Now, the kind of treatment we'll see. It is difficult to treat, especially in severe cases. There are several treatment options available, including medication, salt baths, and quarantine. In medication, there are several ways are available that can be effective in treating this Gyrodactylus infections. These medications can be added directly to the water or administered orally or by injection. Second one is salt baths. A salt bath can help to kill the parasites and reduce the stress on the fish. However, salt baths can be harsh on some fish species. So it is important to research the appropriate concentration and duration of the bath. And the next one is quarantine. Infected fish should be quarantined to prevent the spread of the infection to other fish in the tank. The next one we are going to discuss is diplozoan paradoxum. The diplozoan, it is a unique and intriguing type of monogenean parasite. It's like a dynamic dew in one body. With a visible size of 4 to 5 mm, it catches the eye with its X-shaped body sporting two heads and two hind parts. Its body is bilaterally symmetrical and flattened from top to bottom, with an intestinal cecum present for digestion. Unlike traditional suckers, it boasts a sucker-like structure at its anterior end near its mouth opening, equipped with a sucking disc and an esophagus. The diplozoans has an attachment organ called opisthapter situated at the posterior end of its body. This remarkable structure is made even more efficient by the presence of toluidine blue, a resilin-like substance found in the exoskeleton of arthropods. Now, the life cycle of this diplozoan paradoxum. It all begins when the adult parasite produces eggs that are large, oval, and operculate, measuring 0.1 to 0.2 mm in size. These eggs are then affixed to the gills of a fish by a long thread, and from then, larvae hatch and swim freely through the water in search of a suitable host. Once the larvae find a fish, they mature and become sexually ripened, but not at adult. Youngsters known as diaspora. These diaspora larvae are equipped with a sucking disc and a projection on opposite sides of their bodies, about halfway along their length. In a fascinating twist, each larva inserts its projection into the sucker of another larva, and they grow into Siamese twins that are impossible to separate. These united larvae, known as diporpa, are hermaphroditic, possessing both male and female reproductive organs. They mutually fertilize each other after combining, becoming one flesh in the most literal sense. They are inseparable for the rest of their lives, as they cannot be separated once the union takes place. At this stage, the diporpa have a size of 1.2 mm and the process of combining to form a complete diplozoan is an extraordinary feat that sets them apart from other parasites in the animal kingdom. The symptoms include distress. They become lethargic, swimming sluggishly towards the water's surface in an attempt to gulp air, a behavior known as pipping. This desperate gasping for air at the surface is telltale sign of severe respiratory distress, indicating the detrimental impact of the parasite on the fish's ability to respire properly. Now, how to prevent this? One effective method is to drain the pond and treat the bottom with quicklime at a rate of 2 kg per hectare, allowing it to dry for 2 to 3 days. This treatment can effectively neutralize the parasite and its eggs, ensuring a clean and safe environment for the fish. Alternatively, applying a disinfectant without drying the pond at a recommended rate of 2.5 tons per hectare can also be effective in preventing diplozoniasis. This method ensures thorough coverage and eradication of the parasite from the pond. Another preventive measure is to add methylene blue to the pond water at a rate of 1 gram per 10 cubic meters. Methylene blue has disinfectant properties and can help to control the spread of diplozoan parasites in the pond. Now let us just recall the summary once again. The success of any aquaculture system relies on its design and construction of farm pond and also its proper management practices. The disease management is one of the important parameters in pond management. Various disease-causing organisms, that is the protozoan and the metazoan parasites, are responsible for diseases in fishes. So in this chapter, we had in detail, we had studied about various protozoans and metazoan parasites that cause many diseases. And we also had seen their life cycle, their clinical features, how to control them, and how to treat them also. So water is the medium of living for all aquatic organisms. Hence, maintenance of water quality is a key factor for the successful pond culture operations. So from this chapter, what are the questions that we can expect? Explain in detail about the life cycle, causes, symptoms, and the treatment methods of ichthyobodosis. So we need to mention their characteristic feature, the causes, symptoms, life cycle, and how to diagnose that, and what are the preventive measures, and how it has to be treated also. So mostly it will be for long answer. For 20 marks question, we can expect this. And similarly, write in detail about whirling disease in fish. So same way we have to explain the causes, causative organism, symptom, life cycle, diagnosis, preventive measures, and the treatment. Write in detail about the whirling disease. Then write in detail about ichthyotherius. Write in detail about dactylogyrus. The symptoms, preventive measures, diagnosis, the life cycle, and the treatment. Then write in detail about Gyrodactylus. So with this, we have come to the end of this chapter. Hope you all have understood. Thank you.